Well, good morning. It's a great pleasure to be here. It's nice to come to a meeting where I didn't have to travel too far to go to it. I know you've all come a great distance to be here, and it's a pleasure to talk to you about gene therapy in principle, what gene therapy is, and a little bit about our ongoing gene therapy trials at Nationwide Children's Hospital. I just want to disclose first that I'm a site principal investigator, but have no interest in Aviona Therapeutics, which I'll mention about a trial sponsored by them later. But I am sponsored by, I am a site investigator as well and do multiple clinical trials, mostly in the Duchenne and muscular dystrophy space as well. So with that behind us, I'll mention, here's sort of the outline of what I'll talk about today. I'd like to begin with the idea of genes and proteins. I know many parents are here with new diagnoses, and you all have seen medical geneticists and genetic counselors, but I'd like to begin there in discussions about how we approach gene therapy. Then talk about some of the issues related to gene transfer and, in particular, adeno-associated viruses, the workhorse of gene transfer trials right now, and how some specific challenges related to the mucopolysaccharidoses related to gene therapy and routes around them. And finally, close with some of our clinical trial readiness programs at Nationwide Children's and mention of our ongoing and planned clinical trials. So if this is very basic, please bear with me for a few slides, but I want to make sure we're all on the same page as we talk about things. When we talk about gene therapy, we have to remember that genes, of course, are blueprints for proteins. We go from a chromosome where the gene is resident, and in its several pieces are called exons, and those are transcribed and spliced together into a messenger RNA, an mRNA. And that mRNA is translated by the ribosomal machinery with additions of subsequent amino acids into the protein that does the work, the actual function in the cell. What we're going to be talking about later in transgenes are versions of the assembled gene that called cDNA that is put into a vector that we give to patients, and it's the equivalent of the messenger RNA that we provide in gene therapy approaches. Now, antibodies, there's lots of different types of proteins. Here's an example from the U.S. National Library of Medicine listing some of those functions. And it's important to remember when we talk about gene therapy, there's different ways to do it. For example, there's structural proteins in our muscular dystrophy work. Many of those are proteins that have to be resident in place and are non-secreted, and we have to get them all into the specific right cell. Enzymes are, of course, proteins that carry out chemical reactions. They often, as we'll talk about in lysosomal storage diseases, are involved in breaking down storage products and have some specific potential advantages for thinking about gene therapy. Among those advantages are many enzymes are secreted proteins. So if we get them into tissues that are in roughly the right place, they're secreted locally, taken up locally, or some are secreted systemically and allow uptake by cells at a distance. In autosomal recessive disorders, we know that carriers of mutations for autosomal recessive disorders have 50% of enzyme activity in their blood, and they're effective. So we know that at least 50% can be entirely normal. And in clinical practice, depending on what the enzyme is and what the specific disease, we see patients with enzyme levels of down to 10 or even lower percent who are without symptoms. So we know that there's a wide margin of safety, if you will. We have the advantage with enzyme replacement via gene therapy to get in relatively low amounts and still have a potential effect. As we know as well from patients who've been identified with really low levels but partial enzyme activity who have an attenuated or milder kind of phenotype. So they're attractive targets for the idea of gene replacement therapies. How do you transfer a gene? Well, one way we can think about it is to use just grow up pieces of DNA called plasmids, grow up abundant amounts of them, little circular elements of DNA that contain instructions to transcribe a messenger RNA off it, and inject those into a person. Now, there's advantages to this. There's no limit on the size, for example, of the gene one can transfer using this approach. There's no immune response to it. We could conceivably re-administer it many times, but it's very low efficiency in its transfer. That's why it's really been abandoned. You'll forgive me, I hope, if I use some examples today from the Duchenne field or from the muscular dystrophy field, where clinical trials can demonstrate some of the principles I've described here. And one of them here is way back in 2004, an attempt to use the full-length dystrophin gene, too big to fit in an AAV genome, and injected into the muscle of boys with Duchenne and biopsied 21 days later. And you can see in the picture here, compared to a normal control, that there's really, we're looking at this far column here, that there's really minimal to no uptake or expression of the target protein that we wanted to turn on, that the investigators wanted to turn on. So it's a highly inefficient process. This is where we're going directly into a muscle that we know likes to endocytose or take up plasmids injected in this fashion. So it's not very efficient. That brings us then to the idea of using viral gene therapy. Viruses as envelopes to deliver a payload, to deliver a gene to the tissue of interest. Now, there's multiple viruses to choose from. Among them, for example, is the idea of lentiviruses. Lentiviruses have some theoretical advantages. Lentiviruses they actually integrate into the chromosome. So they place the gene into a chromosome where it's then permanently linked to a chromosome. It can be passed on from generation to generation, for example. There's no significant limit on the size of the gene that one might want to deliver, but it comes with significant risk. Integrating into the chromosome has the possibility of disrupting important regulatory genes. For example, if we integrate into a place that breaks up the function of a tumor suppressor gene, we could expect that giving a treatment like this would cause tumors down the road. So it's not the route of choice. And in fact, that led really to preference for what are non-integrating vectors for clinical development. And the favorite among these non-integrating vectors is the workhorse, adeno-associated virus. It's what's called a parvovirus. It doesn't replicate by itself. It needs to be nearby, needs some of the genes in the adenovirus in order to do its replication. That's why it's called adeno-associated virus. In fact, when we grow these up, we have to grow it up while expressing some of the genes from the adenovirus genome. But it has several reasons that it's favored. One is its presumed safety. There's no known disease association with adenoviruses. There's a whole bunch of adenoviruses, as we'll mention, but there's no known disease association with these. It has wide tissue tropism. Tropism is the word we mean to say it likes to go to different tissues. As we'll mention in a moment, different flavors of this are what we call capsids or serotypes of AAV. Those like to go to different tissues, but there's a wide variety of tissues that can be affected. And that's important for MPS, where we want to consider things like brain and central nervous system, gut, heart, joints, skin, and so on. It creates what's called an episome, a stable, small element of DNA that doesn't get integrated at any significant numbers into the chromosome, but sits within the cell as a stable fragment. And in cells that don't divide, it stays there for years and years and years. Now, some cells do divide. Our liver cells divide. They're called mitotic. They continue to divide. And those cells eventually will dilute out. If you start with 10 copies of this in a liver cell, over time, with each division of that cell, you'll eventually dilute out to nothing. But in post-mitotic cells, it should stay stable for years or even decades. And one example, as we come to the idea of treatment for Sanfilippo or MPS III, the brain is a post-mitotic tissue for all intents and purposes. So if we get it in there, it will be stable and transfect from there for a long time. Although we can say it's not perfect, it does elicit some immune responses, less than many other classes of viruses, but it does. And it has limits to how big of a gene you can get into it. Although, as we'll show you, for our work in MPS III, we can easily fit the entire gene for MPS III genes within an adeno-associated virus. So I just want to go over a little terminology, because I know it's often confusing as you read about this or read about research or different products or things that come out. So we often refer to the capsid or the serotype of this. These are different versions of AAV, and ones you'll see that are in common, are in clinical trials or approaching clinical trials, include AAV1, 6, 8, 9, 10, one called RH74. For each of these describe the viral envelope, the proteins that they differ in the proteins that are inherent in the viral envelope, and therefore they have effects that go preferentially to different tissues with different strengths. So this is important, as we'll talk about, particularly as we think about crossing the blood-brain barrier to get to the central nervous system from mucopolysaccharidosis type 3, as I'll show you some data from. The term you'll often heard too is transgene. So when we talk about transgene, it's the genome construct, or it's the gene that we are delivering within the virus. So then vector refers to the capsid plus the transgene. When someone says they've developed a vector, this means they've got some version of the capsid plus some version of the transgene in it. As an example, a vector that we're doing a clinical trial with now is what's called a self-complementary SC AAV9 with the gene SGSH in it, and then there's a promoter in it, something that tells it to be turned on once it gets into appropriate cells. This is the U1A promoter. So when you hear vector, well, not all vectors are the same. They might differ in capsid, they might differ in transgene. So that's a little sort of background terminology. But it's worth saying that AAV is clearly a maturing technology, and if you go on to clinicaltrials.gov and you put in AAV, gene transfer, interventional trials, you can see that there are 30 active interventional trials right now utilizing AAV. Those are ones that are actively recruiting, and many more that are planned. So we now have a good idea of the safety profile of this in patients as we move forward, and some idea of that it's really going to be the workhorse for the immediate foreseeable future for trials. One such example, I'm sorry, I'm back to the muscular dystrophy field again here, but it's from a disease called limb-girdle muscular dystrophy type 2D, a gene called alpha-sarcoglycan, in which this was placed directly into the muscle. This is the muscle of the foot, just proof in principle of how one can see muscles easy, because as I'll show you when we're done, we can take out muscle and look at expression directly, which is easier to do than thinking about things, for example, with brain. But here, Dr. Jerry Mandel and our team at Nationwide Children's directly injected AAV with the alpha-sarcoglycan gene into a small muscle of the foot called the extensor digitorum brevis muscle. We can directly visualize that it's in there undergoing ultrasound guidance, and it's pretty easy to know where we are. We inject it right in the belly of that muscle, and when we are done, we take the muscle out and look at it in a blinded fashion, looking at a treated muscle and a control muscle. That's what the T and C stand for, compared to a normal piece of muscle. That's what the N stands for. And of the 6 patients I'll show you, there's 3 of them shown here, and we can see that there's a variety of results. In some patients, the treated one goes up with the amount compared to what was there previously, increased compared to control. Another patient had an increase from no expression. These black lines are what's called a Western blot. We're looking at the dystrophin there. It goes from nothing to expression of the protein. And in other patients, in one of the 6 patients, it didn't seem to work so well. But we can see in humans with this and many other, now dating back to 2004, many other trials that AAV gene transfer in principle can work in humans. I'll just mention that this is an example at Nationwide Children's of trials that now we move forward to deliver this instead of direct injection into muscle. We deliver this into the entire limb. These trials are now underway. So what about the mucopolysaccharidoses and challenges for gene therapy? I'm going to share some slides borrowed from my colleague Doug McCarty here, and of course much of the work, in fact all of the clinical work that I'm going to talk about a little later, is derived from observations done by Haiyan Fu and Doug McCarty at Nationwide Children's, who really have developed all the preclinical studies for ongoing gene transfer studies in the MPS at Nationwide. Functional pathways, the lysosomes are sites of protein degradation within cells. There's multiple routes to bring proteins in for this degradation, including what's called phagocytosis. Extracellular material is taken up by the cell and isolated in what's called the phagosome. Endocytosis, large molecules from the cell surface or the extracellular matrix are brought into the cell. And autophagy, where cellular organelles, some of the machinery of cells, are taken up. But all of these lead to what's called a lysosome, where the actual degradation of these products occur. And the lysosomal storage diseases are about 50 rare metabolic diseases with an incidence in aggregate, they're not so rare, about 1 in 5,000 to 10,000 live births. All of them are due to defects of a single gene. They all lead to a deficiency in a specific lysosomal enzyme activity. And all of them are progressive disorders with multisystemic involvement, with a wide variety of clinical features. MPS, of course, is 11 of these, encompasses 11 of these lysosomal storage diseases. And here the tissue of storage is glycosaminoglycan. And there are multiple forms of glycosaminoglycan we'll come to in just a moment. Again, these are progressive, multisystemic disorders. And the incidence of these account for about 1 in 24,000 to 1 in 70,000 births. There are, among the 11, I'm going to talk mostly, in fact, about Sanfilippo syndrome, the MPS 3 category, in which the storage product is heparin sulfate, as opposed to dermatin sulfate or keratin sulfate, hyaluronan. So, in fact, most of our work concentrates on these two, on MPS 3A and 3B, where the gene name that is going to come up as we go in the talk are the genes SGSH and NAGLU, which encodes each of these proteins. Heparin sulfate is degraded in a stepwise process, and each of these enzymes can result in deficiencies of each of these enzymes. Of course, it results in a different form of mucopolysaccharidosis and GAG, glycosaminoglycan storage. The steps we're looking at here are the SGSH step, that's related to MPS 3A, and then the steps in which NAGLU is active related to MPS 3B. But the upshot of this is accumulated storage of heparin sulfate in tissues. This can accumulate in cells, resulting in dysfunction of those cells and ultimately death or degradation of those cells. Those cells clearly are in the neurons and the central nervous system, but also liver and other organs. MPS 3A and 3B are really clinically indistinguishable. The predominant symptoms in these occur and related to GAG accumulation within the CNS, including the brain and spinal cord. I think most of you know children appear normal at birth. Symptoms go through a stereotype course that can really broadly be defined in 3 phases. Usually presentation between age and 1 to 4 years is developmental and particularly speech delay, commonly as behavioral problems. Around age to 3 to 4 years, most patients show clinical and behavioral regression. These include hyperactive and disinhibited behaviors, erratic sleep. And this is the time the diagnosis in our cohort is typically made with observations of organomegaly, liver and spleen enlargement, coarsening of the facial features, and hairiness or hirsutism. And by early adolescence, clearly there's really unrelenting neurodegeneration marked by spasticity, wasting of muscles, and bulbar dysfunction. That's muscles of swallowing and the mouth and face and so on. So our goal, as we'll point out, is to try to, our ultimate goal is to treat patients before any neurodegeneration occur, but our goal in the trials is to treat patients really before entering the final stage, if you will, of this disease. However, we'll point out that, as you know, MPS is really multisystemic. We've highlighted here that Sanfilippo is associated with really cognitive CNS dysfunction, but it is a multisystemic disorder. All of the MPSs are, including cardiovascular disease, respiratory disease, connective tissue disease with joint problems with vision, deafness, GI problems. So our goal in our AAV approach is to deliver it systemically, not just to the brain, in order to be able to address these systemic issues. Now, there's some challenges to gene therapy with MPS, and I'll take them sort of one at a time. And I haven't listed all of the challenges here because there's challenges just in making enough virus to treat enough patients. There's challenges that are regulatory. There's challenges, a lot of challenges to it, but I'm going to concentrate on 4 challenges. One is crossing the blood-brain barrier, as I'll explain in a moment, and different approaches to this. One approach is to go intracranially, deliver directly to the brain. Another is to use viruses that cross the blood-brain barrier. The second challenge is getting enough of cells in the brain to be express the gene to be efficacious, to have efficacy. And this is helped by as you'll see in Sanfilippo syndrome in 3A and 3B, by the use of the bystander effect, which we'll come back to. Another challenge is immunity to the capsid, immunity that exists in the wild, in the environment, and immunity that we generate by delivering the therapeutic vector. And finally, one is immunity to the transgene, when we start to express a gene that hasn't been expressed before in patients. So we'll take these one at a time. So the blood-brain barrier itself is a term you'll hear. It's an anatomic and physical barrier that's formed really mainly by the endothelial cells in the cerebrum, in the brain. These surround blood cells, so instead of the sort of typical leaky blood vessel or capillary elsewhere on your right there, that has really rather loose junctions that allow flow of fluids readily and vector, particularly outside of the capillaries. The blood-brain barrier is formed by structures in the brain, including pericytes, these cells that make a very tight junction around capillaries within the brain, and limit the bulk flow of compounds that can cross this barrier into cells. It's protective of the brain. It prevents most molecules from entering the central nervous system, anything above what's called 500 Daltons, a measure of size of proteins. So it really limits the accessibility of the central nervous system and provides a challenge. One way to get around this, for example, for MPS IIIA, is to deliver this deliver virus directly into the brain. This is a paper from 2014 from the French group, Olivier Danos and others, that shows intracerebral administration of an AAV vector type 10, carrying SGSH and an activating factor in the same vector into 4 patients. And this was delivered 4 subjects aged 2 years, 8 months to about 6 years. And you can see some of the challenges to it. On your left of this picture are 6 burr holes placed into the skull for direct injection of needles, 2 needles at each of these sites. So 12 needles simultaneously injecting into the brain to place the vector there. So in this study, there was no evidence of toxicity, certainly, although you can see even in the images they point out, we can see evidence where there was blood at the site of one of the needle tracks, one of the complications, obviously, about direct injection into the brain. I'll just highlight a couple of things here. In this study, I think an important comment is this, underlined as they point out, efficacy analysis was necessarily preliminary in this phase 1/2 trial on only 4 children in the absence of what would be considered validated surrogate markers. So what can be said about this was brain atrophy. They looked at brain atrophy by MRI, and it seemed to be stable in 2 of the patients and tended to increase in 2 of the other patients. Neuropsychological evaluations suggested some potential benefit, but overall it was not this really demonstrated more convincingly that there was no direct safety signal from doing this approach. But it is an invasive approach with the risk of brain surgery, the risk of sticking a needle into the brain. Another approach is to try, instead of trying to deliver the virus to specific regions in the brain, is to use the delivery system provided by capillaries. Despite this blood-brain barrier, despite this tightness, there is obviously distribution of blood throughout the entire region of the brain. It's enormous. Brain capillaries, there's over 400 miles of brain capillaries, for example, in the rat, with very small brain distances, about 5 milliliters in humans of total volume of brain endothelium that we have to get across. So if there's a way to get across that, it would be a reasonable thing to deliver a systemic agent that crosses the blood-brain barrier to transduce or transfect more of the brain. This is possible by disrupting the blood-brain barrier. Now, this has been shown by a couple of people, including Doug McCarty back in 2009, who showed by using mannitol, which is an osmotic agent, you can expand blood volume transiently, loosen these tight junctions, and get blood flow, increased blood flow out of the blood-brain barrier, allowing improvements in transfer of vectors out of the bloodstream into the brain. However, there's another approach, and that is using the AAV9 virus. AAV9 virus really efficiently crosses the blood-brain barrier from the bloodstream without any additional manipulation or treatment, such as mannitol. This was work from Brian Kaspar's lab at Nationwide Children's Hospital, who showed that an AAV9 vector with a GFP reporter, that's just to show where it went, this is in a monkey, that in 3 weeks after intravenous injection, it quite efficiently gets into multiple areas of the central nervous system, including brain and spinal cord. Here's picture of spinal cord, but it's very efficient at it, allowing us the idea that without further manipulation, we might be able to give the systemic delivery and get it into brain. I mentioned earlier about the bystander effect. So one of the nice things about the lysosomal enzymes is that up to 5 or 10% of the enzymes that are made are actually secreted, as Joe Munzer showed in this figure here from a New England Journal paper in 2004. And these enzymes are frequently taken up by neighboring cells. And if for some lysosomal stores, enzyme levels of perhaps even as low as 1% might be needed for normal metabolic function. But we term this the bystander effect. We don't need to get the gene expressed in every neuronal cell or every central nervous system cell. We have to get it in enough cells that adjacent cells can take it up and have beneficial effect from it. So for this reason, AAV9 has emerged as a vector of choice. I'll point out that there are other vectors. RH74, work by Doug and Dr. McCarty and Dr. Fu have shown that RH74 also is really quite efficient in getting across the blood-brain barrier. But AAV9 gets across the blood-brain barrier without additional disruption. Because of this bystander effect, transfection of a relatively low percentage of CNS cells may result in significant benefit. And the other advantage we postulate is that systemic delivery, by delivering to the bloodstream peripherally as opposed to intracranial, will result in efficient transfection of other affected tissues. Despite the predominant effects being in brain, we do want to get to liver, to spleen, to gut, to multiple issues in the body, which systemic delivery provides us. All right, other challenges. Immunity to the capsid. Well, pre-existing antibodies to AAVs can interfere with efficiency of transfection of target tissue. So pre-existing antibodies to AAV9 can bind to the virus when it's delivered, to the vector when it's delivered, and interfere with its uptake. So these vary in the population. They vary according to age. We know that the rate of antibodies goes up with age just from environmental exposure. So because of this environmentally acquired ones, you'll note that we screen, and everyone screens in gene transfer trials for AAV antibodies, and pre-existing antibodies to the capsid of choice, of the vector, are generally exclusionary criteria from clinical trials. I'll point out that these same antibodies are acquired when we do a gene transfer study. So we are immunizing a child when we give that child AAV. We're giving it, we know that they're going to generate antibodies. So for current gene transfer studies, our consent forms point out that currently we anticipate that retreatment with the current vectors under current understanding protocols won't be a possibility. But I will highlight for you that many groups, including Drs. Fu and McCarty, and many other groups, are looking at ways to get around this by trying to abrogate to treat with medications to get rid of an antibody response at the time of treatment, allowing us the potential to retreat down the road. So we could treat now and potentially treat 5 years later or 10 years later. Although for current trials, our current consent points out that we have to assume we're going to have a one shot to treat this at the moment. Then there's the issue of immunity to the transgene. So our bodies become tolerant to our versions of proteins in utero, and now we're going to provide a protein in gene therapy that's novel to our body as an adult. So this is not immunity to the capsid, but immunity we can induce to the new protein we deliver. So we'll point out that in MPS, many mutations are missense mutations, where a gene is translated into a full-length protein, but a single amino acid is changed and if that amino acid changes a functional enzymatic site, the protein no longer has enzyme activity, yet the protein is expressed with only that single amino acid change. Now, we would expect that in those cases, transgene-directed immune responses would not be seen. They're not expected in that case because the protein has been around, although it's not a functional protein. It's not fulfilling its function. In contrast, mutations that are nonsense mutations or null mutations that result in no production of protein, these would have a higher risk, we would expect, of transgene immune responses. And I point this out only to mention that as you hear discussion of clinical trials, these may be a factor in selecting subjects for initial clinical trials. Depending on what you hear coming up in different places, this might be a factor. And it's one of the factors that helps influence as we think about enrollment of patients for studies. We've seen this, for example, this is my last muscular dystrophy example, I promise, but we see it, for example, in diseases such as Duchenne muscular dystrophy, paper published in 2010, again from across town at Nationwide Children's Hospital, in which a micro-dystrophin version, a miniaturized version of the dystrophin gene was delivered to patients, 6 patients with a variety of different mutations, and patients were biopsied sometime later, at 6 weeks or 3 months later, to look for the effect. And there was no dystrophin expression, but in 2 of those patients, there was clearly T-cell immunity that developed to the transgene dystrophin in 2 of these subjects. So the presence of portions of the protein that these patients never had a chance of expressing resulted in immune responses to it. So that's an example from the real world in which this was seen. So I'll talk a bit about our path to MPS trials at Nationwide Children's Hospital. And I must say that the path has been really a very long one that really began with High-End Fu's first paper in 2002 with Dr. Munzer in North Carolina. What I'm going to present to you in our clinical trials, the culmination of more than a decade of work by Dr. Fu's lab and Dr. McCarty's lab. So I'm going to be talking about, again, this is shared from Dr. McCarty, the current vector genome constructs for MPS gene therapy for 3B, for 3A, and for Hunter syndrome as well. So they look a little different. Each of these shows a portion of what's called the inverted terminal repeats. That's when we talk about the AAV virus, only 4% of the viral genome remains in these vectors that we make, and it's really these inverted terminal repeats that are important for packaging and stability of the genome that we put in. And it was Dr. McCarty who first identified the possibility of making what's called the self-complementary version of these, which are more efficient for replication and packaging of the virus, the viral vector. So I'm going to concentrate on the top 2 of these because the top 2 of these have already moved on to approvals of what are called IND approvals, active INDs. And for your parents who are new to this, IND stands for Investigational New Drug Application. Before we can give an experimental vector to patients, we go to the FDA through a long process and result in an application called the IND. We're assigned an IND number, and only when the FDA approves that IND are we allowed to proceed with the study. So active INDs is a threshold that we know we have to reach that allows us to proceed with the study. So we have 2 active INDs for 3A and for 3B treatment at NCH. This is based on clinical data, again, from Dr. Fu and McCarty. Here's a paper just published in 2016 showing, for example, using the SGSH vector, showing we're looking here at red. I know it's a little hard from the back, but we're looking at the red staining. This NT means the animal deficient in this enzyme without any treatment. And we can see, following treatment, a widespread expression here in liver of the red signal, showing us that the enzyme is made in liver. A little harder to see here in brain. In brain, it's the blue arrows that we show here that show that multiple areas of the brain, multiple cell types take up and express SGSH. And other thing that we want to do, we want to know not only that it expresses the protein, but that it has the function we want. And one way to do that is to look at a protein called LAMP1. LAMP1 is a lysosomal protein that's marked in the accumulation of it marks for us the lysosome, so we want LAMP1 to disappear with treatment. And in fact, that's what they showed. They showed here in multiple animals treated with this that the red staining here for LAMP1 shows a diminishing accumulation of lysosomes in cells. So quite striking results. And this is not only in brain, but in somatic tissues as well, liver, heart, muscle, intestine, so forth, other target tissues, exactly what we want to do in a systemic therapy. And again, in speaking of the brain, our real target tissue, as I'll show you for how we've designed a trial, it shows that there's reduced astrocytosis or scarring in a variety of different portions of the brain, including hippocampus, cortex, and spinal cord, and so on, both in the central nervous system and in the, for example, the plexus, the neurons that help us with gut, that move things through our gut, and so forth. And importantly, clearly, there's an improved survival in these animals that are treated with this. So these are animals treated at 1 month, and you can see a curve here that shows the animals treated with the higher dose, the red line, survive just as long as wild-type animals, animals that are born with the existing gene and with that make the enzyme. It's quite promising. And animals that are treated at different, this one marks lines at different ages of treatment, and we can see that the earlier we treat, the better this survival effect is. So for example, if we wait to 9 months to treat, their lab shows no improvement in survival as opposed to treatment earlier on at the 1 to 6 month age. So this data is actually available if you go online to PubMed, and an article that you can access from home as well. And again, Dr. McCarty and Dr. Fu have shown the same sort of thing with 3B as well. So these are mice injected at 8 weeks and examined at 4 weeks. Just a few more of these pictures, again showing brain, showing widespread expression, both at a lower and a higher dose in all regions of the brain, including cortex, thalamus, cerebellum, spinal cord, and so on. And again, the same issue in somatic tissues. So this gives great confidence that the delivery of the protein systemically results in expression of these 2 genes in regions that we want to do it. Our goal from the clinical side was to develop clinical trial readiness and the clinical trial protocol to do this. We actually began a while back with a natural history study, because one of the issues, if we want to show that something works, we have to have things that we can measure that show a change between treated and untreated patients. So I want to acknowledge first, there's 25 patients who enrolled in a study. Some of those 25 patients I know are here in the room that took part in a study that helped us to do this. We enrolled 15 patients with 3A and 10 patients with 3B. 20 of them completed full visits. As I'll show you, this was a commitment to come back to our institution 3 times over a year for very detailed studies. And our goal was to assess safety of how we measured things and efficacy measures that were relevant to this population. And the results have been submitted for publication. That's what I'll show you now, some of those results. So this was our very detailed natural history study in which we included a variety of measures of cognitive function, a variety of measures under a single sedation event a single sedation event of lumbar puncture and MRI studies, a variety of biochemical measures, and some functional measures as well. And I'll show you selective things from this that have guided the design of our clinical trials. One interesting thing we thought was the anesthesia experience. So issues have been described in settings of MPSs, particularly MPS 1 and 2, about safety of anesthesia, particularly intubation and movement of the latoaxial joint. And we'll note that we had 2 anesthesiologists who performed independent examinations and decisions about sedation, and these 25 subjects were all safely sedated up to 2 times each. And our colleagues, Dr. Cordori and Dr. McKee, are preparing a manuscript sort of to get this out in the literature, that what we've seen with use of sedation in MPS 3 seems to be safe in comparison to what's described with MPS 1 and 2. We found, as expected, biochemical abnormalities relevant to trial design. Elevations in the transaminases, AST and ALT, were elevated. This gray area marks the normal value. You can see blue is MPS 3A patients and red is 3B patients. And it was elevated in a fashion that was up, for example, to 3.2 times the upper limit of normal for ALT. This is important because when we design a trial where we know the virus is going to go to liver, we want to know what ranges are the normal range. We want to know what we can expect for elevations after that. But we have other measures of synthetic function of the liver that were just fine. And I'll point out that this did not correlate with the age of the patient, did not correlate with how big the liver is, and so on. So these are just sort of background biochemical measures. We did spinal fluid analysis, spinal tap, and it was elevated, the protein, a measure of a variety of things, but among them is potentially damage to the CNS itself. It was elevated in 8 of 25 subjects with a suggestion of a correlation to age, but one that's, after we correlate for some red blood cells found in one of the taps, the correlation doesn't quite meet statistical significance, but something else that we'll use, because in our trial, we're looking at levels of CSF protein. We're looking at markers of inflammation in our target tissue. And then looking at urine GAGs, a common diagnostic test, we know that normally diminishes as we go along with an age-dependent range shown here in gray, and we show that it declines over time and almost reaches normal with age. So we used 3 different measures: the lighter scale of a nonverbal IQ equivalent, an age equivalency scale called the Mullen Scales of Early Learning with multiple domains, and a parental reported adaptive behavioral scale. And let me just point out a couple things. One is that the IQ study, the scores decline on mean over 6 months of about 8.625 points, and a reliable change score over that time period we would expect to change. We can measure a significant change of about 6 points over that time period, telling us that this is something we can use to measure changes over 6 to 12 months. But using it for measures less than 6 months is probably not reliable for claiming any significance for our findings. Expressive language clearly was one of the measures that we expect to show decline compared to expected normal development, probably the strongest, along with fine motor over the same period, over a 6-month change. These suggest that changes less than 6 months won't be meaningfully measured by these studies. And finally, I'll say that parental reported behavior using the Vineland scale looks quite good with a linear decline, even up into ages 10 or 11 to 12 years. We don't reach the floor effect of this. So this tells us that we have a range. As our study shows you, we started treatment at age 2, but up into age 7 or so, we still think we can measure a reasonable decline of this. Although this one will point out this, what we call a reliable change score, really implies it will need 12 months to measure it over time. That serves as the background then for the first in-human gene transfer trial, which is now underway, funded by Abeona Therapeutics, in which we intend to enroll 6 to 9 patients in 2 cohorts. It's an open-label trial. The primary outcome is determination of safety of this vector. We have secondary outcomes, which are increase in CSF and serum enzyme levels, reduced liver and spleen volumes at 6 and or 12 months after treatment, improved adaptive functioning over the same time frame, or improved cognitive ability or arrest of cognitive deterioration over the same time frame. These are secondary efficacy outcome measures. Inclusion criteria is age 2 years or older, clear diagnosis of 3A by enzyme levels and genetic mutations, and symptoms, obviously, of disease. We have a whole list of exclusionary criteria, including, I'll just point out a couple of them: antibody titers that exist for AAV9, other medical conditions that would preclude the cooperation or participation in neurodevelopmental testing, other things suggesting CNS or behavioral disturbances that would confound the rigor or interpretation results of this study. Where we stand. So, first I'll say it's dose escalation. So there's 2 cohorts, at 5e to the 12th vector genomes per kilogram, or twice that amount. This is given by an IV infusion over about 15 minutes, and patients are actively followed for 2 years, and their review of records provided for the following 15 years. And this is a terrible slide to read from the back, just to show you all of the visits that are involved in taking part in one of these trials. Is the enormous time commitment on patients and their families. We recognize that and thank patients and their families for taking part. But we have multiple visits. It's a very long and detailed process. So just to give you an update on where we are, we can say in cohort 1, first 2 subjects have been dosed, and we can say clearly that there's no evidence of tolerability issues at present time. And the 3rd subject is enrolled and undergoing screening now. Data from all of these subjects, all 3 subjects, have to be reviewed by the Independent Data Safety Monitoring Board prior to enrollment of a second cohort. And I'll say that we expect meaningful efficacy data, or the secondary outcome data from cohort 1, to really be available at the completion of the predefined 6-month time point in all 3 subjects. That's why we predefined it. We said we can measure this at 6 months. That's when we're going to be able to see a benefit. So that's when we expect to have meaningful data. Presently, we can say after enrollment of the first 3, we're done. So I'll close with that to say, first, I want to just acknowledge a few things. I want to acknowledge, gratefully thank the MPS community, first for participating in our studies, in our natural history studies. Second, for supporting the natural history studies were in fact supported by 3 foundations: San Filippo Research Foundation, Children's Medical Research Foundation, and San Filippo Children's Research Foundation. And I have to just highlight again that all of what we've shown you is the product of long-standing, more than a decade and a half of work from Dr. Fu and Dr. McCarty, and then our whole clinical team that now is involved in these clinical trials. So I'm terribly sorry for running up to the moment of time, but do we have time for questions, or we'll do those afterwards, I imagine, in discussion. But thank you very much for allowing me to speak.